Hello, everybody. Thank you for coming to the seminar today. My name is Russell Galanti. I am the ornamental extension agent primarily for Big Island and also for Oahu. And we're coming to you today from a statewide grant that we've been working on. It's on the viruses and the disease epidemic that's been affecting red ginger across the state. I've been working on this project with a lot of growers. A lot of them are here, and also with the virologist, Dr. John Hu, in the PEPS department, Plant Environmental Protection Services. He's over there, waved his hand, and he's got the beautiful rainbow behind him. And his really incredible PhD student, Adriana Larrea Sarmiento, who's done a lot of the great work on IDing these viruses that I've been tramping across the state collecting. They're pretty much the people we have to thank for all the identification work that's been going on. So how this webinar is going to go is I asked Adriana to put together a presentation on how on the science behind the viruses, essentially, how she was doing identification and our current understanding of the situation surrounding the quick decline symptoms and syndrome that we're seeing in red ginger across the state. After Adriana's done, I'm going to come in and talk about my perspectives from the extension side. It's going to be a lot of repeating, which is good because a lot of this stuff is really complicated and it needs to be said twice. After we're done with my talk, I'm going to ask a panel of growers to come on with me and start to discuss the virus issue. These are people that I've worked closely with on the viruses in either on their farms or if they've shown me virus issues on other farms. And I'll introduce those folks when we come to the panel discussion. It's going to be almost everybody from every island. We're going to be missing Kauai Island because that panelist had a last minute emergency. I want to introduce Hannah Luchin. Hannah Luchin is the extension agent on Maui. I should also introduce James Keach, who's the extension agent on Kauai. We're all ornamental extension agents. Hannah has agreed to help me with co-hosting. So what's going to go on is, unless we ask you to unmute and ask questions, we'll have you just put in questions or comments through the chat. When it comes to the panel discussion, we really want to facilitate everybody talking. So if you've got your own perspectives, ideas, or questions during that section of this webinar, please feel free to either unmute or ask the question through chat. Housekeeping, please stay muted during the seminar so that we can make sure that we can hear the presenters. And if you have any questions, throw them up into the chat. And we'll be putting some information through the chat as well. So I'm going to pass it over to Adriana. Adriana, you can go ahead and share your presentation. I've allowed screen sharing. And while Adriana is doing that, if anybody has any questions, feel free to ask them now.
Hi, Russell. Hi, everybody. So are you able to see my presentation now?
Hi, yes. Right now we see it on not full screen. So it's currently in edit mode.
Okay.
Right now we see your notes section. So we see the presenter view. So you might want to switch over to, if you can switch your screen. Ooh, hide presenter view.
It work now or not?
No, it's still on the edit mode. So if you're on two screens, Adriana, sometimes you can share the screen that just has the full view. So if you have two computer screens going, you can try that.
Yeah, sorry, we recently installed yesterday, so I don't know how to actually put just one screen.
Yeah, you'll get it. We'll figure it out. Take your time. Yeah, so we have been slowly chipping away at this project for the past 3 years, and I think we're all pretty excited to talk about some of the findings that we have. I personally have a lot of ideas for the future of trying to manage these virus issues, and I know there's a lot of growers out there who also have their own perspectives on the issue itself. Some of them I've worked on some of these management options.
What about now? Are you able to see just my presentation?
Yep.
Yep.
We can see it now.
Sorry for the delay.
Don't
So
worry.
welcome everybody. So I'm Adriana Larrea Sarmiento. I'm PhD working under Dr. Young, who is the virologist here at University of Hawaii at Manoa. So we are working together with many people from the CITAR department and also from HARC in the project that involves the flowering ginger and the viruses that are infecting. So before to continue to the viruses, I would like to introduce why the viruses are important for the plant. I mean, why they can cause diseases in the plant. Actually, the viruses can affect the fitness of the plant. Sometimes the virus can induce symptoms, but there are other cases that we cannot observe symptoms in the plant that they are affecting. So, but overall, the viruses that affect ginger can induce morphological and also physiological changes in the plant host. So the main symptoms that we observe with viruses are reduction in the plant growth, that is called stunting, and also we observe discoloration of the leaves, mosaic, and necrosis. But all of these symptoms are observed because we have a change in the plant hormone, as well as a modification of the photosynthesis. So these are the symptoms that we are observing currently in the Hawaiian Islands infecting flowering ginger. So we have a streaking, we have also mosaic, we have a severe necrosis, and also we can observe streaking in the bracts of the flowering ginger. And also we have discoloration in here, as we can observe in this picture. So what I want to also let you know is that there are different ways to infect the virus a plant. So the one is that we call the horizontal transmission. What happening here is that the virus is going to be infecting a vector or an insect, that in this case we have the picture of the aphid. So this aphid could be virus-free. I mean, it's a healthy aphid, but this is going to feed in an infected plant. That is, the plant is going to be infected with the virus. So when the insect starts feeding in the plant, the aphid is going to acquire the virus, ingest the virus, and then is going to be able to transmit the virus to another plant that was before healthy, but now is going to be infected with the virus. So, but all this process is involved the vector in here. So, but we have another kind of transmission that is called the vertical transmission. So what happened in here also is they have an infected plant, and because the flowering ginger is vegetatively propagated, so we are going to use the rhizomes to propagate the plants, or also the offshoots. So in this case, we start cutting the rhizome of the ginger, we are going to start spreading the virus as well. So we are going to have, instead of one plant, plenty of infected plants. So since 2019, the virology lab and the group in here have identified impression of viruses infecting flowering ginger. So in 2009, in Guaymanalo, in Oahu, where the symptoms were observed, the streaking in the leaves as well as in the bracts. So the virus was identified as banana brag mosaic virus. So also the laboratory developed a detection assay. that was very sensitive, but also can be performed in the field because it was isothermal. As now as we can observe the PCR device that were used for the SARS detection, the COVID. So what also they do, they use the transmission assays in using the vector that it was the banana aphid that is called, scientific called Pentalonia nigronervosa. So this aphid was able to transmit the virus to healthy plants. But later in 2017, also there were another symptoms in flowering ginger. Also, the laboratory identified another new virus that in this case was a DNA virus that was called Canna yellow model virus. And also there was established a detection method based on RT-PCR assay that also is currently used for the COVID detection. So, but what happened is we have been not able to identify the potential vector, but based on another literature in the mainland, we speculated actually that the millibug can be the one that is infecting and transmitting the virus to healthy plants. So currently what happened is that we have been observing in Hawaii decline disease in flowering ginger. So in 2018, we can observe here in the picture that there were a stunting in the plants in here in the flowering ginger that before were very tall and high. So in the back part, we can observe the height of the plants that were actually very high, and these plants that were very stunting in here. 1 year later, also there was a report of a dieback, a decline also in the flowering ginger fields, and we could observe here how the plants were dying suddenly. And we have been observing these symptoms also in here at the campus of University of Hawaii. So these are pictures that were taken the last year, and we can observe how the plant was starting showing the stunting, and then now it's showing the decline syndrome or the symptom. So in order to possibly identify if there was a new viral infection in the plants, what we do, we started to performing surveys in the different islands of Hawaii. So during the surveys, we identified in 2019 the presence of a new virus that was identified as a being common Mosaic virus. As we can observe in here the pictures, we have a mosaic in the leaves. Based on the literature, is thought that also the aphid can be the one that is transmitting the virus to an flowering ginger. And also we have developed the detection method for the specific detect this virus. So what happened now is that there are new technologies that have been going on. So we want to use a new technology that is called high throughput sequencing. So what is actually the base of these technologies that we are going to use a plant that is infected. We are going to extract the nucleic acids, and then we are going to run the high throughput sequencing analysis. That is a new technique. And then we are going to analyze in a computer the data that we obtain from this methodology. And then we are going to tentatively or potentially identify the virus, in this case that is related to the disease. So this technology also have been used to detect different viruses, including the COVID. So we use this technology. For doing that, we collect 3 samples that were showing distinct viral symptoms in the island of Oahu. So the samples were collected from Manoa, also for Kahalu, and also the downtown area. These samples were submitted to high throughput sequencing and then we performed the analysis. So what we found so far, these are the samples that were depicting the viral-like symptoms. In here, we have the sample 1, 2, and 3. So among all the viruses that we found and were previously described, that was the banana bread mosaic virus, the Canna yellow model virus, and the bean common mosaic virus. In this sample 1, we found the presence of 2 new viruses that we call virus A and virus B. But in the sample 2, we found 2 new viruses. That was the banana streak virus, the strain Goldfinger, as well as the virus B. And finally, in the third sample that we observed here, more symptoms, the mosaic, we have also the virus B. So what happened now is that among all the viruses, we were able just to identify until now that the banana bread mosaic virus can be transmitted by the aphid. But it's speculated that the bean common mosaic virus and the new virus B can be also transmitted for the aphid. And in the case of the remaining viruses, the Canna yellow model virus, the banana streak virus, and also the virus A, it's speculated that the viruses can be transmitted by the mealybugs. So what we have been doing together with Russell and the different extension agents, we have been collecting samples from the different islands in Hawaii, that include the Big Island, the Kauai, Maui, and Oahu. So in total, we have been collecting 81 samples. So, and this is the viruses. In the top panel, we can see the viruses that we have been detecting. So banana bread mosaic virus was just detecting in 8 samples. The bean common mosaic virus was detecting just in 5 samples, but mainly in Oahu. And Canna yellow model virus, we can see that we have been able to detect in all the islands, and around, and actually 47 samples have been harboring this virus. In the case of banana streak virus, it's not highly distributed among the islands, and we only found in the Big Island and also in Oahu, but just 3 samples. But also in the case of the virus A, is also having detected in the different islands, and 50 out of the 81 samples are harboring this virus. And just for the new virus B, we were able to detect just in the Big Island and also in Oahu. So what we can highlight in here is the virus Canna yellow model virus, the virus A, and the virus B are distributing across the Hawaiian Islands. So out of the survey that we have been performing, these are the virus-like symptoms that we have been identified in flowering ginger. So in here in the A, we have the infection by banana bread mosaic virus, that is the streaking of the leaves. And also we have the presence of the Canna yellow model virus that is going to induce this intervein like yellowing. And when we have actually the virus B and the virus A, we are going to have a misinfection of the virus in one plant, and we are going to have a severe necrosis. But also we have identified the presence of the bean common mosaic virus, and this is the symptoms that are induced for the virus A. And we can see here in the picture of the F that we have a misinfection from different viruses. In here we have 4 viruses that are infecting and are causing severe symptoms. That is the Canna yellow model virus, banana bread mosaic virus, virus A, and virus B. So also what we want to highlight is that there are mixed infections. There are more than 1 virus that can infect the same plant. And what this happens, we can have a severe symptoms, as we can observe in here. In the picture in the left, we have this mixed infection for banana bread mosaic virus and Canna yellow model virus. We have observing that the plant was stunting, also was showing yellowing, and we can observe here the necrosis. While in the case of that, we have another misinfection in the right. In the picture in the right, we have the misinfection for bean common mosaic virus and Canna yellow model virus. The plant was also showing stunting, but we can observe in here the severe necrosis, and in the late stage may be also leading to the necrosis of the leaves. So what we are doing right now actually is a teamwork that is associated, that is involving the CTAR, the HARC, and the PEPs, as well as the Department of Agriculture. So what we are doing first, we want to identify the virus and also to develop a detection methods for the accurate detection of the different viruses that we have. So second, we want to do is study how the viruses are spreading throughout the Hawaiian Islands and the potential association of the viral symptoms with the virus infection. Third, what we are doing first, we want to detect the virus or the potential vector that includes the aphids and the mealybugs. And then we want to perform transmission assays using these vectors in order to see how the viruses are inducing the symptoms in the flowering ginger plants. Four, and very important, we are performing surveys in statewide in Hawaii in order to detect virus-free plants that can be used later for the distribution to the growers. So what we have now, I'm going to go over to the summary point that we have been observing throughout the surveys and the work that we have been performing. So first, currently we have 6 viruses that have been identifying in flowering ginger in Hawaii. There are 2 viruses, that is the banana bread mosaic virus and bean common mosaic virus, that can induce unique mosaic symptoms. However, these 2 viruses, we have been observing that they are not associated with the decline syndrome in the flowering ginger. Also, we have that the 2 other viruses, the virus A and banana streak virus, cannot induce distinct symptoms when are infecting individually a plant. Also, we have that the misinfection by different viruses in flowering ginger are going to induce severe symptoms that are going to lead to stunting as well as a decline in the plant. Another point that we have is the new virus, that is the virus B, is involving in the decline syndrome in flowering ginger. So also we have that we have developed the PCR-based detection method in order to specifically detect, identify these different 6 viruses in flowering ginger. This point that is very important is that the vegetatory propagation using viral affecting material is contributing and may contribute to the rapid spread of the viruses. Also, we have that the PCR that we have been developed to we are using to select virus-free flowering ginger plants in order to perform tissue culture propagation for the potential management of the viruses problem here in Hawaii. And finally, we have that these preliminary results that we have been collecting suggest that the viruses may potentially be transmitted by insect vector. Virus vector identification assays are currently ongoing and be performing here at UH. And finally, I would like to acknowledge all the collaborators in this project that include my advisor, Dr. Yoku, Dr. Wayne Barr also from our laboratory, Shupeng Wang, that is also a member of the lab, as well as Dr. Mike Melser in the agrosecurity lab, Dr. Alejandro Olmedo, that is a PhD working with Dr. Mike Melser, as well as the extension agent, Russell Galanti, as well as Hannah Lactin. Finally, I would like to acknowledge all the Hawaii ginger growers that are present in here and also helping in the collecting and the samples of flowering ginger. Thank you so much for listening.
Thanks for that overview, Adriana. That was good to hear the overview of the science. So what we're going to do now is I'm basically going to say a lot of what Adriana said again, a little bit different probably, and then add a little bit on from my perspective. And away we go. Can everybody see just the screen that is the full screen, not the presenter view? Somebody let me know.
Looks good, Russell.
Okay, thanks, James.
Yes, looks great.
Red ginger disease complex. It's definitely a complex issue, very complicated, something that you're probably figuring out from what Adriana talked about. As a refresher, we've seen this as a reported issue since about 2014. The understanding is that the issue's probably been around for quite a bit longer than 2014, but it wasn't officially reported to H2A and to us, from my understanding, until 2014. This disease complex and the symptoms that we've been seeing first on Oahu and now statewide are resulting in plant health and floriculture production being severely affected. Viruses have been identified as the probable causal agent. So you're going to see probable, may, can, possibly a lot through this talk, and you heard it a lot through Adriana's, and that's just coming with a disclaimer that we can only say certain things conclusively after a certain amount of legwork has been done scientifically. But we do want to give an idea of what our observations and what our hypotheses are for what are the causal agents, what do we think these viruses are doing in terms of symptoms, and what do we think the vectors are. We want to bring what we know, but we also want to give it with a disclaimer that until essentially we've done the scientific rigor, which takes a while, and we've published it, we don't know 100% for certain whether or not what we're saying is true. So you'll know the difference when you hear probable, can, may versus is, isn't, absolutes versus non-absolutes. So from 2018 to 2021, when I was awarded this project and started working with Dr. who's incredible, and Adriana, who are incredible, we've been doing virus identification work across the state. The symptoms we're most concerned with, right, are these quick decline symptoms that we've been discussing and which growers have been seeing. It's stunting of the plants, very quick dieback, necrosis of stems, and in general, seeing the fields being either fields or plants themselves being extremely stunted, closer to 1 to 2 to 3 feet versus the typical 6 to 10 feet that you would see in healthy ginger fields. This is the symptom that we're most concerned about, even though all the symptoms are important because they're somewhat involved with viruses in flowering ginger. This is the symptom that is the biggest issue that we're dealing with. This slide and table is showing an overview, essentially, of what we know of the viruses. And so you're going to see a lot of the unknown possibles on here. On the first column, you see every virus that we've identified so far. There could be more out there, and there could be viruses that we haven't identified. You can see them all the way from Canna yellow mottle virus, banana bract, bean common mosaic virus, banana streak virus, and our new virus A and virus B. And correct me if I'm wrong, Adriana, but virus A and virus B aren't just new to ginger, they're new to science. We haven't seen that these viruses have been identified by anybody else, correct?
Yeah,
and that's why everything's really an unknown with these viruses, because we have to do all the legwork to understand vectors, mechanical transmission, persistence, their prevalence elsewhere in the world. So the second column, we're looking at vectors. We do know for banana bract mosaic virus that aphids are the vectors. For pretty much everything else, we're assuming either aphids, mealybugs, or in the case of virus B, there's the addition of leaf hoppers and plant hoppers. But in general, we're looking at insect vectors of concern being possibly aphids and possibly mealybugs throughout the virus complex. Persistence has to do with how long the virus lasts in the insect vector. And the reason this is important for management is that either a non-persistent, a semi-persistent, or a persistent virus lasts for a certain length of time in the insect vector. For non-persistent viruses, we can try to manage it through preventing the insects getting from a infected plant to a healthy plant in a certain amount of time. That can be seconds, minutes, or days. And if we can prevent non-persistent viruses from spreading quickly, we can sometimes prevent virus spread altogether. Second to last column is prevalence. Adriana showed this, and these are the numbers about the same. It's important to see which viruses we're seeing the most of: Canna yellow mottle virus, virus A, and virus B in terms of prevalence. And then severity of symptoms. This is something we want to understand also for the viruses. So when we're looking at Canna yellow mottle virus, sometimes it's severe. Mostly it's severe when mixed with other viruses. For banana Mosaic virus, first of all, we don't see it a lot, so it's not as much of a concern for us in terms of spread and being prevalent across the state, even though it will cause severe infections mixed with other viruses. Same with bean common mosaic virus, not highly prevalent in our samples, and mostly found severe symptoms are caused when it's with other viruses. Banana streak virus. Rarely see severe symptoms when it's on its own, but yes, severe symptoms with other viruses. Virus A is an interesting one because we found it the most across the state on all the islands. It was pretty much the most consistently observed virus. But oftentimes it doesn't cause severe symptoms on its own. Disclaimer, sometimes it does. Mostly with other viruses, though, it will cause severe symptoms. And virus B does cause severe symptoms on its own often, as we've observed, and it does cause severe symptoms with other viruses pretty much all the time. So that's a good idea, a good generalization of what these viruses are doing. And this is just the numbers in a different perspective to show you where we found the viruses and how much of the percentage of each sample that were infected with each virus. So for example, you see on Kauai, we've only found virus A and Canna yellow mottle virus. That's not to say other viruses aren't there. It's just what we've observed. And 92% of the samples were infected with Canna yellow mottle virus. Oahu runs the gamut. It's almost every virus, with the most prevalent viruses being virus A and virus B. Big concern for growers there. Maui, again, only virus A and Canna yellow mottle virus, this time with virus A being the most prevalent virus detected in samples. Big Island starts to spread out again in terms of the viruses we found there. Found banana mosaic virus and banana streak virus also on Big Island. But you can see those numbers are somewhat low, especially compared to Oahu. Again, with virus A being the most prevalent virus. So it's almost good, in my opinion, that virus A is the most prevalent virus, because if it's on its own, there's the potential that it's not as severe. But of course, if it's mixed with others, we do see often that it's causing severe symptoms. Running through the symptoms again, I'll be quick about this because you heard this multiple times. Banana mosaic virus, we tend to see color breaking in flowers and mosaic patterns. It's always good to repeat. It's hard for me to even remember. I don't remember up until I study the slides before giving the talk, so I'm not expecting anybody to remember these. But luckily this is recorded, so you can go back to this recording. I will send out a video. Canna yellow mottle virus. Sometimes we've seen it where it's just the chlorotic streaking, which Adriana was describing, and we decided we were going to call it vein clearing, because you can see it kind of goes along where the veins are, and the veins are clearing of potentially photosynthetic tissue. And modeling. So modeling is on the left-hand side and vein clearing is on the right-hand side. So this is for a specific plant that's Canna yellow mottle virus positive only. Then you start getting mixed infections. We had somewhere, I think, between 13 and 19 symptoms that we decided we were going to catalog when looking at these plants, because once you start to get to mixed infections, you start to see a combination of symptoms, including necrosis and severe mosaicing, severe stunting. It depends on what part of the plant it was. Sometimes plants were showing it on new foliage, old foliage, stems that were more mature and stems that were less mature. But in general, once you start to see mixed infections, especially for these two, you can start to see how the symptoms get more complex. Again, these are symptoms for Canna yellow mottle virus and bean common and mosaic virus mixed positive, showing the pretty severe mosaic patterns and some stunting. So we're going to talk specifically about virus A, this new virus. These images are all images of plants that we found that were infected and tested positive for just virus A. You can see how insidious this virus is because most of these leaves look like they're not virus. A lot of the plants were full-sized also, and the flowers were full-sized flowers. On the second image to the last, though, you can start to see some pretty specific vein clearing. And here's some of virus A in the field. So on the left-hand side, you can see vein clearing pretty distinctly. And on the right-hand side, you can see a field that was tested positive, but is doing perfectly well in terms of production and growth. That's not to tell people to let their guard down if they were testing positive for just virus A, because it still is serious to have a virus infected plant. And we did see some plants that were just infected with virus A and still showed severe symptoms, even though most of the cases of virus A only infections didn't lead to severe symptoms. But once you start to have mixed infections, like you see in these images, you have severe symptoms from virus A, virus B, Canna yellow mottle virus co-infection, or mixed infections, excuse me, and even virus A and Canna yellow mottle virus mixed infections. So these two on their own sometimes don't show severe symptoms, but at least in the case of this sample, the two combined do show severe symptoms. Doctor Who paraphrased it really well yesterday when he said, we should tell them that it depends on the viruses, the mix of infections, the plant genetics, the environment, and then also plant health. So we know that these viruses will cause the plants to express symptoms differently depending on all of those and possibly other factors. And so that was, again, virus, plant health, environment, and the mixed infections of viruses and plant genetics will be determining how the plants express these. So sometimes you could have a plant that's perfectly healthy and is still virused, even though it's showing some symptoms. Sometimes you'll have a plant that's only infected with one virus and it will be doing not well. And that may be because that it's unhealthy for another reason. Virus B. So virus B was one that we first saw on Oahu. I've seen symptoms everywhere from the typical vein clearing only, where we had plants that only showed vein clearing, up to this more indicative symptom that I've seen from extension. So this is observational only, bleached and distorted foliage. So oftentimes the leaves are showing a bleached, as if they've been sprayed maybe with Roundup, or if they've been sun bleached, like scorched, but not yet necrotic. And then also distorted foliage. And most of this is on the upper leaves from my observations on newly emerging stems. Here's some images of what it looks like directly in the field. So if you're looking down on your plant and you see this, you potentially have virus B. Here's another plant that tested positive for just virus B, and you can see how severely stunted it is, even though it only has one virus infection. This was what one grower was saying was Polynesian princess. So this was a plant that was also showing color breaking. Whether or not that is a result of the virus, we don't know for sure. General conclusions from IDing. Ginger plants presenting mixed infection with the new virus A, virus B, in combination with Canna yellow mottle virus, are sometimes associated with decline of the plants. Virus A is highly prevalent, but does not result in severe symptoms alone, oftentimes, usually does not. Virus B can result in severe symptoms, particularly in mixed infections with other viruses. And usually mixed infections with 2 or more viruses leads to severe symptoms. I just want to talk about fungal and bacterial pathogens because these were of concern when we started the project. No bacterial pathogens were identified in initial samples of decline symptoms. We pretty quickly decided that we thought bacterial pathogens were not likely to be the causal agent. Many fungal pathogens have been identified throughout this project. Dr. Miaoying Chen was the mycologist that focused on this project with us. You can see on the right-hand side how many different types of fungi she isolated during sampling. It was a ton. Most of those fungi turned out to be the ones you see in this list. I don't want to name them because nobody's going to remember the names anyways. The most important we want to remember is Nigrospora and Phoma. These were the most prevalent. Both of them were found in over half of the samples that we sent. We tried for pathogenicity. That is a method for determining whether or not fungi can actually infect plants and whether or not they're the pathogen that we're seeing. It's called Koch's postulates. You have to do a series of steps to determine whether or not these fungi match with these symptoms. Unfortunately, we were not able to identify pathogenicity or prove pathogenicity on Nigrospora or Phoma, even though they were the dominant fungi. Nigrospora did cause lesions in some of the pathogenicity tests, but we weren't able to consistently cause pathogenicity. That being said, probably the symptoms we see on this image right here are associated with Nigrospora. It's yellow lesions that can often lead to becoming more necrotic. You can see that on the right-hand image, and you can see the beginning to mid stages of the yellow lesions on the left-hand side. So without Koch's postulates, we can't say for sure that these are the symptoms that are being caused by Nigrospora, but these were the symptoms that we were able to recreate by infecting some plants with Nigrospora. So possibly, I'm saying probably, the symptoms are associated with the fungi Nigrospora. So we're going to talk prevention now. This is probably what everybody's most interested in. We're deciding we're trying to start with virus-free plants. We've done some testing with trying to infect plants with the vectors, the insect pathogens. And we also know that these viruses can be spread through propagative plant material. So if you start with a virus plant and you propagate it, you're even cakey or from rhizome division, you're probably spreading the virus unless it was a brand newly infected plant. So vegetative propagation of virus-infected plants will nearly guarantee you start with a high viral load. Most likely the virus has been spread statewide due to vegetative propagation because that's how everybody's distributing ginger, giving it to friends, or repropagating it for their farm. That's not to say that insect vectors aren't playing a role, which they most likely are, but we're almost guaranteeing we're spreading virus by propagating virus plants. So do not move infected plant material and possibly sanitize your equipment that touches infected plant material. It's really hard to tell everybody to disinfect your equipment every time you cut a ginger or every time you divide a plant. But if you're cutting ginger in infected fields, you are better safe than sorry using some sort of sanitation protocol for pruning your ginger or cutting them for harvest. We have virus-free plants. So we started to move to look for virus-free plants after we determined that that was probably the best bet in terms of getting ginger production going again. So you can see the insect exclusion cage that I labored over for days. It's got an insect exclusion mesh screen that thrips can't even get through. And we've got virus-free ginger sitting in that cage, slowly growing. We've given some plants, thanks to Hannah, and thanks to Adriana for testing them, to myself, and Oahu has them at 2 locations, the Hawaii Ag Research Center, whose waiting to do tissue culture once we get funding, if we get funding. Hopefully we get funding at some point. And then Adriana's got virus-free plants as well. So there are 3 locations where we know we've got virus-free plants when we tested them. Of course, we're going to have to test them again. This isn't a perfect system. An aphid could have flown in there right when I opened up this flap to get a picture. So we always have to retest to make sure they're virus-free. So remember when I was talking about persistence, non-persistence, this was an original recommendation I had made for dealing with non-persistent pests, intercropping, barrier cropping, and density of ginger plants. Aphid vectors can transmit certain viruses instantly, the non-persistent ones. Aphids generally colonize edges first, so you can possibly use border crops to clean aphids of the virus. This only works for aphids and for non-persistent viruses. If you have the willpower and the ability to do this, using barrier crops or barrier cropping your field by making it in an isolated area where there's no other gingers or potential hosts, which is difficult because the potential hosts are probably around, you could try to protect your crops just by isolating them or adding a barrier crop. Scouting your crops, your barrier crop, to determine if you have pests before they enter your field, and then possible timing of chemical applications through scouting. You would have to scout weekly though to keep up with insect life cycles. Cultural management. So we can try to keep healthy plants, and sometimes we do see that plants are more healthy with still having either single or mixed infections. That may be genetics, it may be environment, but there's potential cultural management strategies that we can employ to try to manage our healthy gingers. Avoid excess fertilization. Gingers are heavy feeders, but excess fertilization like high nitrogen can encourage aphids, mealybugs, and even fungi. Mulching and increasing organic matter is time and time again proved to be a boon to crop production. Maintaining soil moisture, that means keep your gingers wet pretty much. At first, I was thinking it was not a good idea to keep them wet because of fungal pathogens, but at this point, when we know the fungal pathogens are playing a role in ginger decline, but they're probably not what's causing the plants to reach the decline stages, we do want to keep our plants well watered. Plants, in my observations, under shade seem more tolerant to being infected with viruses than plants in direct sunlight. In all the fields that I've seen, without knowing all the fertilization, without knowing what the organic matter is in the soil, I saw that plants that were in the sun and plants that were too dry were the most severely affected plants in terms of plant health when they were infected with viruses. On the bottom table, it's showing recommended foliar nutrient levels for ginger production. This is in the publication that I sent to everybody who signed up for the webinar. And if you want the publication, just email me afterwards and I can send it to you if you didn't get it. Biological, somewhat small slide. We have recommendations for non-chemical pesticides, organic pesticides. Refer to the publication or ask me. Again, higher soil organic matter in many research publications equals better disease suppression. One thing that we did want to look at one point, but hasn't been done, is using chitosan to try to prevent fungal pathogens mainly, but it can also work against insect pathogens. Chemical. Fungicides are useful and have shown effectiveness to keep ginger plants healthy. One of the first ginger fields I saw that was infected with virus but was looking healthy was a field on North Oahu. The farmer swore treating it with Manzate always got rid of the issue. That's to be determined, but he certainly didn't have any fungal infections in his field, and he sprayed mainly with Manzate, which is mancozeb. But a good rotation good fungicide rotation is always recommended. Insecticides, broad spectrum insecticides aren't recommended because how they affect beneficial insects. Also because we don't 100% know how effective they are. If some of these viruses are non-persistent, you can't spray enough to prevent them from entering your fields. You would have to spray too consistently for most growers to be comfortable with. But you can refer to the publication for pesticides because the spraying is an option to reduce insect vectors in your field. Soaps and oils are good recommendations because they're not going to kill broad spectrum pests and beneficials. Of course, support beneficial insects for aphid predation. One area of the chemical applications that would be interesting to look into that there's no work on in gingers, but there is elsewhere, are anti-feed oil. So these oils reduce transmission by preventing aphids or other sap-sucking insects from feeding. They essentially touch the oil with their mouth part, and they decide not to pierce the plant tissue because the oil is unappealing. Some of those oils are labeled APH oil, JMS stylet oil. So stylet is the aphid's mouth part. And you can see the recommended intervals to spray 5 to 7 days and anytime it rains. So anybody in Hilo is probably not going to be happy about that, and anywhere where it rains. And then there's anti-feed insecticides. So these are more harsh insecticides. Right now, they're not labeled for Hawaii, or the only one that is labeled for Hawaii is only labeled for veg crops. But just for your knowledge, those two are the ones that I found as of this presentation, fulfill and belief. Other options. I've talked a lot with growers. I talked with Eric during the last couple of weeks about what if we grew under shade or grew in insect exclusion screen and had spray programs. Or I've talked with others, what if we grow in isolated areas? And this is where I want to take part of the discussion today. I want to acknowledge everybody that Adriana acknowledged. They've all been very helpful, along with, let's see, who do I want to thank the most? Hawaii ginger growers. Thanks everybody for letting me come to your farms, for allowing us to do sample collection. It's meant for the best of the industry. We don't give away the information. We just want to try to figure out the issue. And so thanks to everybody who has participated and allowed us to take samples, because it's really led to the understanding that we have now. And if you haven't let us come out and now you're interested, then please contact me. So where we're at now is I want to invite the growers that I asked to be part of the panel. We've got somebody from every island. Unfortunately, Johnny Gordinez is not able to join us. He had an emergency, so we don't have anybody from Kauai, unless there's somebody who wants to jump on and be the Kauai panelist. Maybe James, you can point somebody out or ask them privately. So I want to invite Adriana back up. Dr. John Hu, who's going to be on the panel. So he's the virologist. He pretty much helped me make sure that the presentation was accurate. And he's the guy that's kept us knowledge base wise on track for this whole project. He's a wealth of knowledge, and I'm sure he'll have some comments. I want to invite Portland Law, who's got a ginger farm on Oahu. So I dealt during my tenure on Oahu a lot with Portland's farm. She's a really good grower who's interested in taking care of the land well. She was close to the epicenter of the pandemic, as you would, being close to some of the first farms that tested positive for the virus, and we had reports. I want to invite Shane Castillo, who has a hardy amount of ginger down in Ka'au. He's a grower that we worked with a little bit to a lot on his gingers. And he's somebody, sorry to out you, Shane, who tried the barrier cropping. So Shane gave the barrier cropping an attempt. Who else do we have? Allison Wright. Are you there, Allison? So Allison's also a grower.
Yes, I'm here.
Yeah, hi. You're a grower. You're a landscape management manager, and you've got a lot of experience with diseased gingers on Maui. I know so because we've done a lot of virtual walkthroughs of Maui infected ginger. So to start off, do we want to run through the questions, or would everybody like to introduce themselves? Actually, yeah, let's go through and do maybe 2 minutes each grower, starting with Portland. Your experience, 2 to 3 minutes of your experience with the virus and your biggest takeaways. And then if you've got something that you want to talk about with other growers, go ahead and share that.
Sure. I'll go first. I was hoping to hear everyone else first, but we moved to our farm in 2018, and we primarily had red ginger and heliconia varieties growing here when we came here. When we moved here in the summer of 2018, the gingers were beautiful. They were tall, they were green, the flowers were big. And over the course of a year, I watched slowly, very slowly, as the height of our plants, specifically more on one side of our property than another, were slowly over time getting smaller and smaller and smaller. A lot of the pictures that you saw, we were seeing it all over our farm. We were seeing the yellowing, the striation on the leaves. We were just seeing everything. So that is when I reached out and met Russell, and he came out to the farm and took samples. And we've tried a lot of things. Farming red ginger turned out to be a lot more work than I expected. It's not as easy as just planting something and watch it grow and harvesting. I see all of you nodding. So we've learned a lot, and we could definitely be doing a lot more. We've done mulching, organic compost. I've been doing compost tea, not as regular as I should, but we've been doing a lot of compost tea spraying. I've actually started some barrier kind of things, if you will, and we redid a lower field, and we did rows of red ginger and 2 rows of ginger and 1 row of heliconia to kind of separate them. And we noticed a lot of success in the health of our ginger plants when we did that. Yeah, that covers everything. And now we're at 2021. We're still dealing with it. Now we've got plants of all different heights. Every now and then we'll get a taller red ginger, but we do have plants that are clumping a lot smaller than they should be. But we're pulling through. My goal is to try and save the plants instead of get rid of them. A lot of our neighbors have just left the red ginger farming completely and switched to other crops. I'm hoping that over time we can succeed in bringing them back to life.
So I had a question, Portland. Did you ever get shade trees into a portion of your farm?
You know, we planted those ones that you brought. They actually didn't survive. I don't know why. But I've started some other shade trees along the edges. But yes, shade is a huge help. The heliconia, the taller heliconia shading the gingers have really helped.
Yeah.
But yeah.
So we'll go east. So we'll hit Allison next. How about that?
Hello, everyone. So my name is Allison. So I've been in the landscape industry on Maui for 18 years. So it's very different. So Russell said I'd work in floriculture too, but that's a different part of what I do. But for the landscape industry, so it's very different working in the landscape industry than in the floriculture industry because of different ways you're growing everything. So we can't cover our plants. So But over probably the last 10 years is when we started seeing plants start to decline, but it wasn't everywhere. It was sporadic around different properties. So I work everywhere from Wailea. I'm in Hawaii. We work up country. We work on the west side. So we have, properties all over Maui. And when ginger plants get old, they start to get stunted. So we were thinking, oh, they're just old plants. We should divide them and just to kind of regenerate them. People started saying, oh, look, we have dwarf gingers. So we're like, no, they're not dwarf gingers. So it's kind of going through a process of trying to figure out what was going on. But what we've done is after we've seen it, it tends to be, if the issue is on a property, it's really bad. So I have a few properties that are aware where they know, your ginger plants are a foot tall. So versus, 6 feet tall, which what they used to be. So people are very eager to hear about this, or most of the time people just don't know. So with all the landscape industry, people don't know what's going on. They don't know the issue. They just think that's how it grows. We've had some success with changing the varieties. If a client really wants a ginger, we'll put instead of just an Alpinia preparata, we'll put a Kimmy Pink ginger in, or a Jungle King ginger, and they do a little better. But we've been recommending to switch to heliconias or switch to the shell gingers, which are another Alpinia, but different species. And those don't seem to be as in much of an issue with us. So, but I do see mostly the fungal spots, and that tends to be kind of like the end stage of everything. And that's when your plants are only a foot tall. I do know, because I do a lot of plant purchasing. Most of our nurseries here are pretty good. I know a couple people who have plants that they look like they're kind of virus free to me without testing them. It's just a visual thing. But I have noticed some of the box stores bringing plants in that are notably have issues. But it's a question of, what do you say to them? Who do you go to say something without definitive proof to go tell a manager, hey, can you not bring this plant in because of this issue? So, and that's
Sure.
kind of how we're dealing with everything here. So if anybody has questions about the management, there is nothing we've been really able to do. So that's an issue in the landscape industry.
Yeah, a lot of the samples that we collected on Oahu were also landscape plants. So they were people calling us in Waikiki or Honolulu, or a higher end management companies were asking us to check their plants out. And having virus infected plants in our landscapes and in our home gardens is definitely a prevalent source of infected material. And we did test positive plants in nurseries on pretty much every island also.
And I did ask a few tissue culture labs on the mainland that I buy other plants from to see if either they grow any kind of ginger or if they'd be interested in. And it's really not something people have thought of yet or have put in their plan, because I've told them, if you have anything that's virus free, if you can guarantee me it's a virus free plant, that I'll buy 5,000 of them and bring them over. And but
Sure.
it's just changing the issues like that. And like I said, with nurseries, everybody here on Maui, and specifically all the nurseries, they're just redividing their plants and reselling them. So
so we'll follow up on a little bit of that afterwards. We'll let Shane Castillo know over as far as we can get over here on Big Island. So Shane, maybe you want to talk a little bit about your experience when you first noticed it, what you've been doing, because I know you're really motivated to handle this issue on your farm and on others.
Hello, everyone. There's too many variables and a lot of unknowns as to where we're going, what we're going to do. But just speaking, plain English, farmers, we don't have a lot of money to deal with this issue. So we have to find mitigation somewhere in between to see what we're going to do. And I think the best solution in my own personal trials that with the fields that I have, and even with the newly planted stuff, is age is a factor of the plant. Second thing is plant health. Dr. Hughes said it, or told you, Russell, that the phenotype is has a direct result of the plant health in way of the form of the gene of the virus expressing itself, and are different factors now where photosynthesis play a vital role, whether it be in the shade, whether the plant makes more photosynthesis, whether it doesn't make more photosynthesis. So there's too many variables, and so trying to understand where do we go from here? What do we do? We can pull out everything, but how fast will we be able to replace? And if we are washing and preparing for shipment inter-island anywhere between 7 to 1,000 gingers every week, you can only imagine how many plants there is to replace. So very motivated into seeing what possible mitigation solutions that we can all come up with.
Sure. Thank you, everybody. I want to first address we've got a couple questions in the chat, so maybe I'll just ask it to us all. Dr. Hu, Adriana, any growers that want to jump in. This isn't just for the panel of growers. Anybody can talk about their concerns, their thoughts on it. So I know Eric Tanoe is here. Eric, feel free. You and I talked the last couple of weeks on what you thought about growing for yourselves, and we've talked a lot about it over the past 3 years. Colleen asks if anybody has recommendations or suggestions for good aphid deterring border plants, I would appreciate it. I'm not going to answer that. I'll go first. I'm not going to answer it from a deterring border plant. I will answer it from my understanding of good border plants for ginger. You often want to try to find a plant that isn't a direct host for the virus that we're dealing with. We don't know for these viruses what all the plant hosts are. Some of them are new to science, like we said. Some of them, we just haven't done enough testing to understand who the hosts are. But it would be good, and if it was me, I would try to find a plant that's as distinct from ginger as possible, as far away in terms of evolution as possible. But it also has to be fast growing, taller than the gingers, something that's probably a windbreak. So my first and foremost would be a plant that's a windbreak. You want a plant that's tall enough and thick enough that it's not letting wind through, and it would also prevent insects that are either flying or caught in the wind from entering easily. So essentially a plant that's like a net. It also should be fast growing because you got to get it up quick. It has to be distant evolutionarily from ginger. One that I've recommended in the past has been Podocarpus, and pretty much any of the plants that you're thinking of in terms of windbreaks that we use right now are good windbreak, are good border plant options. So Cook's pine for the biggest. Heliconia is one that is a concern because it's somewhat evolutionarily close, but it is also a flowering plant. So if you're a cut flower grower, it's a plant that you can grow, that you can still harvest from, that isn't ginger. And we haven't found that has been experiencing virus symptoms, even though we often see Heliconia grown intermixed or very close to ginger. Those plants aren't suffering from viral infection symptoms like we see gingers are. Anyone else? John, Dr. Hu.
Very good, Russell. I think your answer is very good. I just want to add a little bit more about the aphid transmission of the viruses, because that is an important issue. Both Russell and Adriana talk about that, but that is also a complicated question because we are talking about different viruses. As Adriana said, those two potiviruses, banana bread mosaic, and the bean common mosaic, they induce very unique mosaic type symptoms. They suppose they are transmitted by aphids. But from what we have observed, I've been talking with Russell and others, seems like those viruses actually in the field are not, based on what we have observed, are not vectored much by the aphids. However, one of the two new viruses, the virus B, we don't know, but based on the nature of the virus and other similar viruses, that one probably is vectored by aphids, probably. And that virus is very different from banana bread mosaic virus and bean common mosaic virus in terms of transmission. Remember what Russell used the term non-persistent, persistent? And this virus B probably more than likely is what we call persistent. That means it takes a longer time for the aphids to acquire and to inoculate the viruses. So if that's the case, whatever you can do to control the aphids probably will be helpful to at least reduce transmission of the virus B. But I have to tell you, to control the aphids, to control the spreading of the potiviruses is not a good idea. But the good thing is, again, based on what we have observed, seems like the aphid transmission of those two viruses is not very common, at least not very important. That's what we have observed. Now, the next question about transmission, can the viruses spread through soil? Well, based on what we know, some of the viruses, but not the ones we have identified, are spread out by soil. So I guess my simple answer is no. As far as remove the severely infected plants, by all means, yes, because those plants, they look ugly anyway. They do not produce good flowers for you anyway, and they serve as a virus source anyway. So if you have enough time, labors, get rid of them, by all means, that's good idea. Yeah. Well, there are more questions now. Let me give back to Russell for now.
Sure. Okay, so James over on Kauai has been asked about the color breaking in Polynesian princess. This is a plant that a lot of people hold near and dear because it's one that was advertised as a plant that was this candy cane striping cultivar called Polynesian princess. And sometimes it's sold for much more than your typical ginger is sold for. And they ask, and I've wondered this myself, is the color breaking solely caused by the virus, or is it independent of infection? That's tough to answer because we don't have the original Polynesian princess. We don't have any plants called Polynesian princess that are not infected with the virus. So we can't say for certain without having the same genetics of the plant, but unvirused, because we just simply can't find it. I personally, from my observations, believe that Polynesian princess is a color breaking cultivar because it's infected with the virus. All plants that we have tested that are called Polynesian princess or growers think are Polynesian princess have tested positive for one or more of the combinations, mixed combinations of viruses. So I'm way more confident that it is caused by virus because we see color breaking as a symptom of virus infection, not just in ginger, in other plants as well. So I always like to just say, if it was me, I would assume my Polynesian princess was infected with virus. We can't say for certain, but more than likely it is a viral symptom. What do you think, John?
I agree.
Yeah.
I agree. And also, as far as I know, at least we know a couple of the viruses we have been working on flowering ginger. They do cause color breaking. Yeah. And even the banana bread mosaic virus and the one Russell showed you, that's a very clear color breaking. Yeah,
Sure.
on the flowers. Yeah.
And so we saw one with Canna over in Hana that was a cultivar called Peppermint. And it was basically Polynesian princess, but a tall plant. The plant looked healthy. The flowers were almost normal size. I could tell from the foliage that was highly mosaicked and from the flower that basically looked like a peppermint candy in how much color breaking it had. And of course that tested positive for the virus A and for Canna yellow mottle virus. So there's other cultivars out there that are being sold as these color breaking cultivars, but more than likely they are plants that have been infected with one or more virus and it's showing color breaking as a symptom. So if your plants are small, if they're stunted, if they're color breaking, they're probably not a cultivar, they're probably a virus plant.
Russell, can I say more?
Please.
Okay. Now, again, thank you guys for your help, allowing us to collect samples from your farm so we can do the research. It's actually frustrating for you. I know when we identify so many viruses from the disease plants, and we still cannot tell you, many of you know other virus diseases like papaya ring spot virus on papaya, banana bunchy top virus on banana. It's very clear. We know that one particular virus on papaya causes that disease. One virus on banana causes bunchy top. But apparently on flowering ginger plants, now Adriana has been working on 6 viruses, and we may still have more. So again, different viruses, different combinations on different plants and different environments may induce different types of symptoms. So it's very challenging for both you and us. Now, one thing I do want to emphasize, Russell has been trying to get some funding support to produce clean plants through tissue culture, working with Hana and others. I think eventually, even we don't know a lot right now about the nature of the viruses, about the transmission of the viruses, but I still feel for this crop, for this problem, probably the clean plants, to start with clean plants, by all means, is the way to go. So I was telling Russell the other day, for other crops like apple, grapevine, potato, sweet potato, people have been trying to produce clean plants, sponsored, supported by the federal grants. But for us in Hawaii, this kind of small crops is difficult to get the federal grants to work on this kind of projects. But collectively, with your support, hopefully Russell can get some support from the state government. And he has a wonderful, wonderful team, including the growers, the industry, the researchers from UH and PBARC, and also HARC. So I think Russell is a good leader for you guys on this project. Yeah, thank you all. Thank you, Russell.
Thanks, Dr. Hu. To follow up on that, yeah, so we've got a legislative request in for this fiscal year. We're asking for $120,000. It's a new project request that we've given to the legislature, and it would cover trying to produce virus-free ginger, and it would cover funding a graduate student to work on management strategies for dealing with the virus. And that's what we're looking at for the next phase of dealing with this virus issue. We think from our experience that the best thing to do is produce virus-free ginger, at least while if we can get more funding to fund research to determine more details of what the viruses are doing to the plants. At least we know we need to get virus-free plants out to the industry. And that's our best conclusion right now. That's our best understanding of management. And from a statewide big picture perspective, that's probably the best way to go about managing the viruses right now. If we can produce a lot of plants, smaller growers, newer growers, and growers that feel that they have the ability to replant their virus fields will have the opportunity to do so. And instead of spreading virus plants through vegetative propagation over a long, slow period of time until it becomes a huge issue, we'll be able to start virus-free.
Russell, can I add a little bit more? Sorry,
Sure.
I talked too much.
No, you're fine.
Not for every virus problems we will give you this recommendation. For example, for papaya rispa virus, I won't ask you to grow healthy papaya plants if they are not resistant, because simply they just got infected too fast.
Mm-hmm.
And even for banana bunchy top, I won't tell you to spend lots of money to propagate bunchy top free banana seedlings for growers, because the aphids just spread the virus too fast. And before you can have your money recovered from harvest, and then your plants will already be reinfected. But based on what we know so far, I feel, again, based on how much we know, at least I want to echo what Russell said for your flowering ginger virus problems. Using clean plants is the way to go. Yeah, thank you.
So I just want to bring one other comment up, and it was when I was talking with Eric Tanoe last week about how to grow the plants, and this would be another strategy. Him and I were, and this is for you, Dr. Hu, it's for other growers who think they may have some understanding of the issue or how to grow along with the other extension agents. Eric and I were saying, okay, if somebody's a grower and they've got shade house, for example, or they can build shade house easily, or another sort of greenhouse structure, would it be a good production style to start to grow ginger under shade cloth? At least that's a slight preventative in terms of netting against insect pathogens, with a pretty consistent spray program, or even adding insect screen either to shade cloth structures or in replacement of shade cloth structures as a way to produce ginger in a more production style. So we're talking first a structure with shade cloth or a shade cloth insect exclusion net combination along with a spray program. My first understanding is that, yes, the more protection you have from insects getting in, the better. It needs to be fully enclosed, right? Shade cloth's good, but it can't fully enclose against something like small aphids or mealybugs. They'll still get in over time. So you would want to also use a spray program. Insect exclusion cage is good, or insect exclusion screen is good, but it's expensive. And it gets damaged. So any new hole can cause an insect to get in. Also, people tend from observations to see that once you have insects that enter an insect exclusion area, they run rampant because there's also beneficials that are excluded from the insect exclusion zone. So there are some considerations with this growing style that possibly it could work in my mind. Possibly there would be some issues that you would have to iron out later. What do you think of anybody about, this idea of growing either in exclusion zones or under shade cloth with a heavy spray program, or not so heavy, a spray program essentially? And we're talking growing in rows, so you can either walk down the rows or you can drive equipment down the rows. This would be something where you have to start to grow in a more conventional ag design, in my opinion.
Russell, Addison has an interesting question about the viruses on tulips. Yes, actually, I use that as an example in my virology class. On tulips, if they are infected by, actually, they are similar viruses as papaya ring spot virus, banana black mosaic virus, bean common mosaic virus. So if those plants are infected by just the potiviruses, because those kind of streaking or stripes or color breaking, they're kind of actually kind of unique.
Yeah, they're really attractive.
But the whole plants are still relatively normal. But Alison, the problem for us is you have so many different viruses, and particularly the new ones, the virus B, and the combination of different viruses. So it's not just color breaking, it's the whole plant, as you said, they get stunted and decline, even eventually die. So it's not just color breaking, not just stripes, not just streaking. Yeah, if it's just those color breaking, maybe it's kind of a unique. Yeah.
Well, that and I used to grow a lot of tulips when I lived in Pennsylvania, and we did a lot of tulip farming. And the color breaking flowers that were infected with viruses, we always would throw them out every year versus saving them, because those tulips often didn't flower again well enough the year after. And it depends on which ones were virus. Some we tried to keep around, but oftentimes they were in a state of decline because they were virused. So yeah, you can use it from an ornamental perspective, but for a plant that's perennial, continually growing like ginger, I don't think it's a good idea, because we haven't really seen color breaking that has been associated with also very good production.
Hey, Russell.
Yeah.
So in my opinion, I think we're kind of heading to a world in floriculture that there will be no such thing as clean, virus-free plants. There's going to be so many virus.
Mm-hmm.
And because there's a lot of liner trays that are being created out of micropropagation methods, and we've got all these cool different kinds of variegated leaves that they're now propagating. And so I think I think like soil health, is there any way you guys be able to maybe collect some data on soil pH, maybe the possibility looking at, I know that Adriana talked about imbalance in plant hormone, maybe expounding on a later session as to what that might be and what that might consist of. Also looking at maybe photosynthesis in all places where the data was collected, just to get more information to kind of narrow down what the best mitigation method might be. Because right now, getting rid of all the ginger plants in the state of Hawaii is, I don't think so anyone will agree with that.
No, nobody's gonna be able to do that. So how I would see the next phase going is that we successfully get funding, right? We've got a portion of the grant that goes to virus-free production, because first and foremost, I have to re-emphasize that if people can plant virus-free and start virus-free with their production, this would be new growers, this would be growers with new fields, not necessarily trying to replant everything, but those that can fill the gaps in what is a void of ginger production across the state. That's going to help. We need virus-free plants. A lot of the variegated plants that we see, I can tell the difference between a variegated plant and a variegated plant because it's virused. And we need to manage that. Even if people are spreading around, it's not good to just say, well, it's going to happen, because we need to make sure that it doesn't. And Hawaii is a really, really susceptible place for that kind of thing, because we always get plants in. Okay, so part of the grant, about 60 grand, is going to go to HARC, right, for producing virus-free plants. We're going to do as much of that as possible. The other 60 or so grand is going to go to a graduate student. What I would say is that to that graduate student, they would do a literature review to try to determine what the best avenues for looking at plant health and looking at management strategies are. So we're going to leave it up to the graduate student, along with a committee, myself, and a graduate chair, to guide them to determine through a literature review and through our expertise what experiment they should set up. So what parameters in the plant, soil, environment they should look at, and then what treatments they should try. It's going to be not a huge project, because we're only looking at about $60,000. Hopefully it promotes the development of other research projects. Once we publish it, people across the world can also work on research. So we haven't published yet and let the world know, other than a poster session at a virus conference, that we found this many viruses in these gingers, and a few other publications, of course. But it's going to be important to tell other people and in other ginger producing areas of the world that this is an issue. It could be an issue for them if they haven't noticed, and hopefully promote research. So it's going to start slow with an initial graduate student project, and then from then on, we need to keep going. And of course, we'll reach out to everybody that grows ginger to see what their thoughts are.
Shane, as a grower, I'm curious what mitigation strategies you've used on your farm, and if you've noticed anything in particular that's really helped, or have noticed any recovery.
Sure. So I'll let other growers answer first, but Portland, you've got a neighbor close to you, one of the first people that showed virus infection on their farms. And when I went back and talked with him, he said his gingers bounced back completely. No. And have you noticed that they've stayed healthy since then?
His or mine?
His.
I have not been over to his farm. I've only I would like to. It's giving me hope that someday that mine might magically
bounce
back. But
so I've heard miraculous stories of people who have been severely infected with ginger. Their whole field has come back after dying back, and it's been regular sized. Now, this is something that I have observed in the field, that at one point they were infected and then they looked very healthy. That's something that is unexplained right now. And I know Portland, you've worked a lot on organic options. Yeah. And so you've said they've kind of not worked that well. It's mostly been shade, correct? That's
actually what works really well is that organic compost with a layer of mulch. It is just so expensive. It's not something that is economically feasible for us to continue like every 6 months. Mm-hmm. And we even use some of our own wood chips, but it's not enough to cover our fields. And I can attest to like having shade or like shade screens, something like just shade in general is really beneficial for the plants. But yes, we've only tried organic methods. And with the organic manure compost that we've done on some of our plants, I have noticed a visible change in the like overall health of the plant. The leaves are much greener, striation patterns disappeared. One thing that never recovered though was the height. It wasn't like, these plants were all of a sudden getting all the nutrients they needed, just bouncing right back up. But overall they looked healthier, but the plant itself was still small. The flower is still small. I have tried the heliconia and rose in between ginger, and that's worked really well. It's provided shade for the gingers on either side. And those are a really fast growing plant. They are a cut flower, so it is beneficial in that respect. But you do get the height and the shade relatively quickly. to cover the gingers. One thing I've done now in one section of the farm is I've used chickens to actually manage and underneath the flowers, the weeds and the bugs. And I've noticed plants becoming much healthier. Again, we're not getting the height that we would love, but I've noticed that as an interesting method. I don't know if anybody has thoughts about that, but just having chickens maintain the ground of fertilizing it, keeping it clean and weed free, eating those bugs I have noticed a benefit to my plants that way.
So I had a question that came from the comments. Where do you get your compost from?
Where did we get our compost from?
Yeah.
I got it from a friend, just a connection he's a landscaper and he knew how to get organic manure compost. I've contacted several horse farms, but I haven't been successful in securing like a good contact to get compost from.
Sure. So anybody else?
Sorry.
No,
I didn't
go
know
for it.
about the sanitizing of tools and things. We did try that for a while. I was sanitizing instruments when we'd harvest in between each plant. I'd rotate out of a bucket. I've used, we were using bleach. I just noticed that the tools just corroded and like they were unusable after a week. So, really
bleach is the least recommended for any of the corrosive metals. Neo asked if anybody's used Virkon. Virkon is known from research to be successful with disinfection. So any of your labeled disinfectants, like Virkon, any of the Clean Grows or Green Shields, all of those that are labeled as disinfectants for tools and for greenhouse surfaces themselves. Hannah, did you add that publication into the chat?
Yes.
Yeah, so Hannah put the publication on sanitizing. We wrote that. Oh, right? That's ours. Yeah, we wrote this publication. So take a look at the publication on sanitizing tools and greenhouse equipment and everything. And you didn't hear it from me because it's not labeled, but rubbing alcohol's really good. So any of the alcohols are really good for disinfecting equipment. They just aren't technically labeled for it. But nobody's going to come down hard on you for using those. I use them. I will say it on recording right now that I use it. Let's see. So does any other grower or anybody want to talk about their experience with trying to mitigate the virus?
Hi, Russell. Can I talk a little bit?
Oh, please.
Yeah, I don't have too much experience because I don't grow flowering ginger in the field, but actually I was, because the project started and we don't have anybody that actually had the experience to grow flowering ginger in here. So I struggled a little bit in the beginning trying to grow flowering ginger, but thanks to Hannah also, he was the one that helped me in the beginning to establish the ginger plant. But what I realized, at least at the greenhouse, is that the soil actually is very important for the plant health. So what we are using, at least at the greenhouse, and I can maybe recommend to you, is maybe you have a plant that is looking healthy, just put in a pot and try to isolate maybe 10 pots, because flowering ginger is going to replicate very fast. So maybe you can just put a very small screen upon these 10 plants that are going to be your source of plants. But basically we're using is the potting mix soil number 4. And actually we use a very tiny amount of the slow release, the fertilizer. And just always we put under the shade. The shade, as all of you mentioned, actually is helping a lot with the plants at the greenhouse, at least at the greenhouse, to be more healthy. And if you put under a shade house, is going to help also to protect from the insects to spread the virus. But I think basically, if you put in a greenhouse, We bought one very small from Amazon that was just $60 or $70. That was a greenhouse, a portable greenhouse that you can buy in Amazon. So that has doors and everything. So you don't just need to assemble very fast, and that will help you actually to put the plants in a protected area, and maybe that can be also your source of plants. Thank you.
Thanks, Adriana. So final thoughts, because I know myself and a few others have a meeting at 5. I've noticed statewide, plants have been healthier when they've been in the shade. Plants have been healthier when they've had good soil to grow in. So good soil organic matter, the right amount of fertilizer in the soil, and plants have grown better in areas where they're receiving enough water. So I tend to see more issues in virus plants in some of the less rainy areas of each island. So that was on Oahu and Maui. I mainly saw this. In drier areas, the plants were affected more severely from the same types of virus infections. These are just observations. This is nothing that I've tested statistically. It's just my observations. And that's it. Shade, soil health, the right amount of fertilizer and water have been observably beneficial to infected plants. I think we need to wrap it up because I know we're way over, but I do appreciate everybody sticking around because I'm sure it's very important for you. Hey, so I'm here. Email me if you want to talk more about this. You've met some of the individuals. If you want contact information of other folks that you'd be interested in talking to, it's important that growers help each other through talking about and helping manage their issues. If we keep quiet about everything, the industry as a whole is going to suffer. So let's practice helping each other out. So thank you, everybody, for coming to the seminar. Thank you, everybody, for talking. Thanks, Portland, Allison, Dr. Hu, Adriana. Shane's already gone, but thank you, Shane. Thank you, Hannah, for co-hosting.
Thank you so much, everyone.
Thank you.
Thank you, everyone.
Bye.
All right,
Have a
folks.
nice weekend. Bye-bye.
Bye.
Once this is up on YouTube, I'll send a recording to everybody that signed up for the webinar. Have a good evening, everybody.
Same to you.
All right. Take care, y'all.
Thank you. Great job.
Thanks. Thank you. See ya.